I think network issue. Good afternoon, everyone. Welcome to the fifth day of our research methodology lecture series. Okay. Good afternoon, everyone. Welcome to the fifth day of our research methodology lecture series. I hope you enjoyed yesterday's session. Today's session on the topic of randomized control trials, RCT and their types, which is going to be presented by Dr. Renu Rathi ma'am. She has completed her BMS, MD in Comma Nritya, and had fellowship in health professional education. Also done a web course of bioethics Haifa, and SWAM course in fundamentals of clinical research by ICMR. At present, she is a professor in Department of Comma Britain Mahatma Gandhi Ayurved College, Hospital and Research Center, Salod, Wardha. She has a total experience in teaching of 21 years and for PG teaching of 5 years. Ma'am, she has a 20 years of experience as HOD. She presented 26 papers in national and international conferences. She worked as organizing chairman in 8 seminars and as an organizing secretary in 5 CMEs or workshops. Her publications are 100 for international and national indexed peer reviewed, 31 in Scopus PubMed, 8 in Web of Science, and 34 in others. And 10 papers are under publication. She completed 30 projects with IAC, 8 by self and 24 with scholars. She also completed one funded project with government collaboration MGIRI. She received 4 copyright and 11 are in pipeline. She got National Best Teacher Award of ATA in 2020, Best Paper in National Conference, Best Essay and Video in National Competition in COVID-19. Her achievements are: she started Swaneprashan in Maharashtra in July 2010, appreciated by Lawyers Club for Health Services through Ayurveda resource person in Ayush CME at Raipur on Comma Nritya. She is a PG guide, guiding 6 students, 6 in progress, and as a PhD supervisor since 2017. 5 scholars are registered under her supervision, and 1 scholar is awarded PhD degree. Ma'am has a clinical experience of Ayurveda practice for 22 years. Now I would like to welcome Dr. Renu Rathi to present today's session on the topic of randomized control trial and their types. A very warm welcome, ma'am. Please start your presentation.
Sound बढ़ाना चाहिए। Hello.
Hello. Shall I proceed, Dr. Pitosh? Hello, Dr. Pritosh.
Yes. Yes, ma'am.
Your voice is much echoing. Am I audible and visible? Hello. Shall I proceed?
Yes, ma'am, you now proceed. You are audible clearly.
Okay. Good afternoon to everyone, my dear participants, maybe faculty, PhD scholars, and PG. Very good afternoon to all. Please listen carefully that this topic is much needed and have great demand nowadays because our city is a gold standard and everyone must thoroughly go through it. Now we will today we will see the type of research, definition of research, key elements and categorization of clinical trial, what is bias and what are errors in design, then different types of study designs, enlist the types of control, randomization clinical trial and its classification. Then we will see advantages and disadvantages as well as do's and don'ts. Whereas we will see again the checklist or certain guidelines that enables us, that help us and facilitates our research as per the proper guidelines to maintain the GCP guideline and high quality RCT. Now we will see what is research. You all know that there is a journey from known to unknown is research. There is a quest of learning is the mother of all knowledge and the method which man employs for obtaining the knowledge for whatever the unknown can be termed as research. So in short, research is refers to the systematic method consisting of formulating the research question, then framing a hypothesis, then collecting the data, analyzing the facts, and reaching certain conclusion either in the form of solutions towards the concerned problem. So the objectives of research are to gain the familiarity with a phenomenon or to achieve new insights into it. Such studies are termed as exploratory or formulative research studies. Certain studies are described the particular individual, time, place, and situation or a group are known as descriptive research studies. Whereas certain studies determine the frequency and contributory association with which something occurs is known as diagnostic research studies. We can name it as a diagnostic as well as prognostic research studies too. To test a hypothesis of a causal relationship between variables, such studies are known as hypothesis testing research studies. Now come to the types of research. There are so many types of research. So one by one we will come across with what is the exact meaning of that particular research. we just see that descriptive research includes surveys and fact findings inquiries of different kinds. As the name itself suggests that there is no control group or no any comparison. Whatever facts are coming, whatever facts are we are collecting, that are only need to represent. So there is a no variable, no control over the variables, and we have to report what has happened or what is happening with no comparison group is the characteristic of descriptive research. In analytical research, on the other hand, the researcher has to use facts or information already available, does not assign the exposure and analyze these to make a critical evaluation of the material. For example, cohort, case control, and cross-sectional. In these studies, there is a comparison. We have two groups, and we have comparison of these groups, and this can be prospective as well as in cohort we can use as a retrospective as well as mixed also. If we talk about applied research, then it is a finding a solution for an immediate problem facing a society. For example, COVID-19, we have faced a big problem of COVID-19, and in that way we can do research as an applied basis. Regarding fundamental research, we need to add certain information or generalization with the facts as a formulation of a theory, and it is an add-on information as a basic research. So the main aim of applied research is to discover a solution for some pressing practical problem, whereas basic research is directed towards finding information and that has a broad base of applications and thus adds to the already existing organized body of a scientific knowledge. If we talk about quantitative versus qualitative, then the term itself depicts that it is based on the measurable outcome. Quantity itself, the term denotes as a measurable outcome. It is applicable to phenomena that can be expressed in terms of a quantity. Example is anemia. Here we can quantify hemoglobin pre and post treatment. It involves the generation of data in quantitative form, which can be subjected to rigorous quantitative analysis in the formal and rigid fashion. And its subtypes are inferential approach, experimental approach, simulation approach. Now come to the qualitative research approaches. Here it is the concern with subjective assessment of attitudes, opinion, as well as behavior. And the results generated are in non-quantitative forms, in the form of gradation as a mild, moderate, severe, like that. If we talk about conceptual versus empirical research, conceptual research, the term itself suggests that it is related to some abstract or ideas or theory, some concept, new concept, or to reinterpret an existing ones. And while empirical research means it relies on experience or observation alone, often without due regard for system and theory. It is data-based research and coming with conclusions which are capable of being verified by observation of experiment. And it is experimental type of research, and it has most powerful design and possible for a given hypothesis. Here we can test the hypothesis. Clinical or diagnostic research. Such researchers follow case study methods or in-depth approaches to reach the basic causal relations. Such studies usually go deep into the causes of things or events that interest
Ma'am, you are not audible. I think some poor network issue.
This conference will
Okay.
now be recorded.
Now we will see types of research. Clinical or diagnostic research. Such researches follow case study methods or in-depth approaches to research the basic causal relations. To reach the basic causal relations. Such studies usually go deep into the causes of things or events that interest us and with very small samples and deep probing data. Exploratory research are meant for development of hypotheses, and formalized research is studies are those with substantial structure and with specific hypotheses to be tested. So method techniques or and methodology means systematically solve the research question. Research question where research methodology means we need to solve so many queries like why, what, how, in what way, or where, and like similar other questions, and need to solve our research topic. Need to reach our research topics. Research topic must be very sound enough and scientifically merit updated as per suggestions included with scientific research committee and have medical utility as well as commercial value. Practical consideration and organizational factors should be added and make it enough competent and sound. We can take help from so many research engines like PubMed, Metacrawler, Dogpile, Profusion, ZepMeta, or Google Scholar, and also peer-reviewed literature, original articles, review articles, as well as textbooks. First of all, we need to define the research problem, then review the concepts and theories and previous research findings, and accordingly, we need to find the gap analysis, we find the gap where the uh, previous study designs and new our research question as per gap analysis and formulate the hypothesis. Then we go to the methodology, thorough methodology and strong appropriate research design. Then we need to collect the data and analyze and interpret the conclusion. So in this way we have to conduct our research. Now come to the different variety of study designs. Study design, clinical research aimed at evaluating traditional medicine should incorporate the conventional concepts of research design, such as RCT or other types of clinical studies as a observational study. And we need to stick on certain guidelines like CTRI or GCP, good clinical practice, and consort. We will see one by one. So, what are the study design types? Mainly, there are two main types, observational and experimental. Observational, again, mainly it includes no exposure not manipulated by investigator. Here, investigator have no control over the exposure or any uh, intervention. And observational can be divided into analytic as well as descriptive. Analytic means there is a analytic in analytical study design, there is a control group, means comparison is there, for example cohort and case control. And in descriptive design there is a no control or no any comparison. Examples are case series or case report and cross sectional or ecological study design. Experimental studies are interventional studies, also called as clinical trial, and in this there is a exposure manipulated by investigator. There is a fully control over uh, investigator. What is the actual meaning of clinical trial? Clinical trials are revised schedule by of the Drugs and Cosmetic Act 2005. It is a systematic study of new drugs in human subjects to generate data for discovering or verifying the clinical, pharmacological, or adverse effects with the objectives of determining their safety and efficacy of the new drugs. But in Ayurveda, there is a no as such new drug. Already, we generally take the intervention from the our Sanhitas or from known. But if we have to research on folklore medicine, then we need to we can treat as a new drug. Key elements of clinical trials. PICO, everyone knows PICO. P for patient or population or problem. I for intervention or therapy or test or diagnostic tool. Whatever exposure is going to occur, then it is called as intervention. We need to compare our outcome with other group as a comparison or another therapy or placebo or other standard control trial. Then O for outcome means result we need to check whether which one is good one. Then we need to plan a good and sound foundation of successful clinical trial, because failing to plan is a planning to fail. So our blueprint, our protocol or synopsis must be good enough and a design must be strong. Then only we can conduct a good research. Now coming to the how to categorize clinical trial. There are two mainly participating as per the participating centers, like single center or multi-center studies. We know that our faculties or PG, PhD scholars do only single center study, in which only single center is there. But we maybe we need to do multi-center studies, all different zones to be added from east, west, north, south, as well as center, from center zone of the country, and we can make the population as a randomized control, and then we can put our research forward and conclude the results. Also, as per like from all the zones as a national study, and where if we take samples from different continents, then it can be as a international study, and where we need to take approval from a regulatory board. Whenever there is a single centered study, we need to take approval from our institutional ethical committee as the IEC. But when we conduct the research from abroad, and it is an international status, then we need to take approval from regulatory board. We can eliminate the subjectivity by adding, by removing bias and errors. What is bias? Bias is a systematic error contained in the study design, conduct, or interpretation of a study. Whereas extensive list of particular bias forms exist, there are two main basic forms of bias: selection bias, information bias, and we can so many the types of biases are there, like confounding bias or outcome bias are very important bias, in which if we don't consider or don't eliminate it, then our study may not be strong or the outcome will be shuffled. So we need to remove it or eliminate it by information bias or confounding bias. For example, confounding means if we have to undertake the research on Tribhuvan and still there is a paracetamol is giving, someone is giving paracetamol as a co-intervention, then paracetamol here act as a confounding bias, because already we need to check the result of fever of Tribhuvan, and if we are giving paracetamol as a co intervention, then the results are confounding, because there are two antipyretic drugs are going on, and that's why the results are confounding. And outcome basis, if there is a outcome bias, means outcome bias if we uh, if a participant know that this I am going to assign for this particular trial or this particular intervention is going to be given to me, then if it is a trial, then he may exaggerate the thing that the results are I am suffering with this and that. But if the trial is already proven or standard control, then he may hide the things and may not report. So there is a outcome bias. Now we will see control group. Control is an important group in which we need to compare the results of trial group. So control groups are so many, like negative control, blank control, or positive control, placebo control, then reference control, standard control. So negative or blank control, what is the meaning? The meaning itself suggesting that it is a no, as such no intervention is being given to participant because it is a blank or negative. So no intervention is being given. There is a no production of results because no intervention is there. While in positive control, there is a result is expected because we are giving certain intervention. Then third is placebo control. Here placebo is an inactive substance in which we can think that the results may arise due to psychologically. Because participant don't know about the inactiveness. So psychologically he may get results. Blinded control or double blinded, we will see one by one what is the meaning of double blind, single blind later on in further slides. Randomized standard control or standard therapy. Randomized control means we will see what is randomization. Randomization means every participant must get chance to assign the or enroll in the study. That is called as randomization. And standard control means there is a already proven results. Suppose paracetamol is a proven drug for fever, so we can take it as a standard control. And if we need to take, we need to conduct study on Tribhuvankirti, then Tribhuvankirti may be act as a trial group and paracetamol as a standard control. And what is reference control? So reference control is suppose one good formulation is given in Bhaishajya Ratnavali. Then Bhaishajya Ratnavali reference, we can take it as a reference control because they are much authentic. The formulations given in 52 authentic books are declared as per the standard, and we can take it as a standard reference control. So how to remove, how to eliminate, or how to avoid biases? What are the techniques to eliminate them? The two most important techniques are randomization of the subjects and blinding of subjects as well as investigator. Then monitoring should be conducted. Monitoring should be there for every clinical trial. Thereby, no as such manipulation of data or as such no misinterpretation of data not be done. Checking original source documents and source verification, data verification is important. And also there is a need to check the quality control as well as quality assurance of procedures. Now we will see randomization. It enables the equal chance to being assigned in any of the groups and separates the factors other than intervention. Common methods are non-randomized. Whenever there is a no randomization, we can call it as a CASI experimental design. CASI, QUASI, CASI experimental trial, when there is a no randomization. And when there is randomization, there are 2 to 3 major types of randomization. One is simple randomization, second one is blocked randomization, stratified randomization, or cluster randomization. You must need to know that strata, cluster, cohort, these depicts as a group. Strata is a group, or cluster or cohort is denoting as a group of people. So whenever we talk about stratified or clustered or cohort, then the whole society or the whole ward or one whole community, that particular area is considered, and the two groups, two clusters or two stratas or two groups are being checked or being assessed for that particular intervention. So blinding, or what is blinding? Blinding means concealed allocation. We need to blind the things, blind the intervention, so that one cannot exaggerate the things, or one could not hide the or misinterpret the things. So there is a different stages or methods of blinding. One is open label, means no blinding is required. Whenever we talk about herbal herbal intervention clinical trial, then there is a much difficulty to get to hide the things or to make it blind, because there is a no blinding. The major prerequisite for blinding is uh, the groups must be very identical in look, color, and taste, and everything must be very identical, means similar. And that's why if we do not able to make them similar, then blinding could not be possible. And so the trial is open label, where there is a everyone knows what is being assigned or what is being given. So it is called as open label or non-blinded trial. Then single blind design. Single blind design means where participant don't know about about intervention, what is being done, what is being given to him or her as a intervention. So it is a whenever patient or participant or subject is not known to the intervention, then it is called a single blind design. When patient or subject or participant as well as researcher don't know about the intervention, then it is a double blind. And the prerequisite is the trial medicine, the trial intervention as well as standard control medication intervention must be very identical and similar. So no one can guess that which is trial drug and which is control drug. Now third one is triple blind design, where subject, investigator as well as statistician or sponsor don't know about the intervention. Now coming to the types of clinical trial. There are three types of clinical trial. One is superiority trial, where if we assume that the outcome is superior for the trial outcome, interventions outcome is more good than control or than comparative standard control trial, then it is called as superiority trial. In inferiority trial, where alternative therapy or where the trial outcome is not much good enough than standard control, then it is a inferiority trial. And where we expect the result, the outcome is similar from that of comparative from that of placebo or standard control as well as trial group, then the results are when equivalent, then it is a equivalence study. Whenever we assign the exposure, then it is called as observational study. If investigator assign the exposure, if exposure is there, then it is a experimental study, and when there is a no exposure, then it is a observational study. So experimental study can be divided into two again. When there is a random allocation, then it is called as RCT. And when there is a no randomization, then when open label, then it is called as CASI trial. Where comparison is there, then it is called as analytical study, where we can divide it into three, that is cohort study, case control study, and cross-section study. And this all three has comparison, comparative group, two groups are there. That's why it is a analysis, which one is good and which one is bad. So here analysis is being done. So with the help of comparison, comparative group, it is called as analytical study. Where there is a no comparison, then it is a descriptive study, like survey. Whatever finding is we get, then we need to represent it. So it is a descriptive study. There is a no comparison or no any control group. Classification of clinical trials. As per purpose of trial, the purpose are different, like treatment trial, therapeutic trial, or prevention trial, or screening trial. Like COVID-19, we need to screen whether the patient is having symptoms of COVID-19 or not, like that. If we need to improve the quality of life, then the trial's name is as a quality of life trials. Compassionate use trials or genetics trial. If we need to conduct the study on genetics, then it is a genetics trial. This is the example of sample of placebo controlled study design. Here the placebo is the trial group is probiotic honey, and the added with the bacillus coagulans as a probiotic, and compared with plain honey without addition of lactobacillus. Here randomized control trial on Ayurveda interventions with nicotine chewing gum for de-addiction of tobacco products in school-going children of Salod Savangi, Bardha. In this study, there is a Nicotex chewing gum is a standard control, while Ayurveda intervention as a withdrawal trial is a trial group. So the two groups are there in which we need to compare the result outcome with the help of proven uh, intervention. Example of prevention trial. A new vaccine has been developed that has safety and ability to produce immune response against that particular disease is a prevention trial. Plan 1 is just for 10 to 15 or few people more than less than 50 or less than 30 is a phase 1 trial. Then progresses to phase 2, 3 and 4 respectively. Then randomize participants into two groups as trial and placebo with randomization. Then we need to conduct follow-up periodically to record safety and efficacy. The main characteristics of RCTs are randomization is must, then size calculation is must, and whenever we talk about clinical trial, the size is not based on population, but it is based on previously proven sorry previously proven results. It is called as effect size. It is based on uh, previously result of SD. What is SD, or what is the expectation of results like 10%, 20%, 30%, and what is the expectation of our new research design, new research question, and as per the uh, difference, if it is difference is equivalent, then it is equivalence trial. If the difference is less as compared to previously proven result, then it is the inferiority trial, and if we expect the results more good enough, then it is a superiority trial. We need to take consent. Informed consent is very must in randomization trial. Then we need to approve the results, approve the synopsis or protocol from institutional ethical committee. Then we need to, our methodology, our methods should be strong enough to avoid the bias and errors. Type 1 error, type 2 error, you already know about it, what are the errors. We will not go in detail about errors because the otherwise it will go so much lengthy. Then any new intervention we can check with the help of RCT, like vaccine, new drug, or new diagnostic kit, like that. Randomized control clinical trial. It is a planned experiment designed to assess the efficacy of prophylactic, diagnostic, therapeutic agents or devices, procedures, etc. applied to human subjects. Here human is very important because it is a clinical study. Essentially, it involves comparing the outcomes enrolled in a prospective study. The study must be prospective. It is in future we need to check the results, and retrospective means already we know we know the results. Then it is a retrospective. If we want to check the results of from last 10 years, what are the percentage of cancer patients in that particular hospital, is a retrospective study. And prospective means we need to check in future what are the outcome of the study, then it is a prospective study. Exposed to intervention in control and trial groups followed over the same or double period of post treatment, means after completion of trial, after as the treatment get finished, then we need to check we need to call the patients as a follow-up after post-treatment follow-up up to the period of same period of treatment or double period to check the residual effect. What are the advantages, disadvantages of RCT? Advantages are, this is a gold standard, evidence-based, and has ability to update the science with scientific facts. Whereas disadvantages may be complex and expensive, and it needs immense ethical challenges. Then it lacks representativeness or no choice of group selection, because it is a randomization is there. But participants could not um, uh, enroll himself or herself with his or her choice, and there is a need to pay compensation as per calculated by DCGI, that is Drug Controller General of India. And advantages are, uh, it is a controls confounding bias without adjustment, without any manipulation. It facilitates effective blinding in some trials. It can, we just discussed that double blind, single blind, triple blind, like that. It can be possible in RCT. It maintains advantages of cohort studies too. So these are the advantages. Now, randomization means certain community, certain patients, the population is here. We need to check, we need to divide into two groups. Here we have to make randomization, and this is assigned to one group and this is assigned to two groups. And we can ensure that both the like black and white, this both characteristics should be equal as per the population. Means everyone must get chance to enroll himself. That is called as randomization, and it is a gold standard of evidence-based medicine. It is essential to control various known or even unknown biases. What are the fundamentals of randomization? Researchers must be unable to predict the group to which a patient will be randomized until the patient is unambiguously registered on study. So he has no right or no selection choice to enroll himself. Then researchers must be unable to change a patient's allocation after they are randomized. Once if I am getting the result from control group only and not getting result from our trial, then I could not shift them from trial to control and control to trial. So researchers must be unable to change a patient's allocation after once they are randomized. Interventions of two groups are exactly identical and in double-blind RCT design, and not to know the drug allocation of both, thereby elimination of subjectivity in judgment of outcomes can be confirmed. What are the techniques of randomization? Fixed allocation randomization, we already discussed this. Then adaptive randomization and response adaptive randomization. This is the example of simple randomization, that is computer generated table. It is taken from www.randomization.com. And if we have to assign two groups, multimodal therapy as well as Ayurveda intervention, then it is randomly or haphazardly comes as a two for one first number and second number two multimodal. Then only third number is single one is Ayurveda intervention. Then again multimodal. Then consecutively four enrollments of Ayurveda interventions. Like that, it is not a serially or ordered allocation, but it is a randomly allocation. So it is a as per generated table, computer generated table, we need to assign the patients into two groups. What are the hello? What are the benefits of randomization? It ensures the assigned treatment cannot be predicted at the time of patient selection, and treatment is independent of the patient's characteristics. Variables likely to affect the study are distributed at random, means it prevents bias, and statistical analysis based on the type of randomization should be planned with the help of biostatistician. We have already discussed this. Advantages and limitations of blinding. It is logistically, limitations are mainly, advantages are protects from assessment bias, and it checks placebo effect, psychological component too. But limitations are logistic complexity, ethical complexity, blinding in procedure based therapies. Blinding is difficult in practical or impossible things. It is difficult to keep herbal interventions as a blinded. Using active and control formulations with similar color, taste, and weight, treatment blinding may be possible. We need to arrange the identicity. Then only it is blinding is possible. Factors to consider in selecting a clinical trial design: study population and indication. So treatment duration, we need to consider these factors. Thereby we have to decide whether which design we should undertake. Treatment duration, carryover effects. Many times patient come with us after taking so many medications, and later we, in a chronic basis, he come to us. So there is a carryover effects of previously taken medicines. Then it is a cost and logistics. We need to think upon budget also, and if budget is feasible, then only we have to go forward. Then patient convenience and statistical consideration. It needs statistical biostatistician. Then only our by statistically we have to check the whether results are statistically significant or not. Running period. What is running period? Running period means there is a no there is a some period lacking, period between the previously taken medicines. We need to keep some gap, and later on we have to start our trial. It is the between period is called as running period, and the advantages are it acts as a washout period to remove effects of previous therapy. It can be used to obtain baseline data and to evaluate if patients fulfills inclusion criteria. It can be used as a training period for patients, investigators, and their staff. It helps in identifying placebo responders, and it provides useful information regarding patient compliance. Now coming to the placebo controlled clinical trial. The placebo is given to an add-on treatment to an already existing regimen that is not sufficient to treat patients. The placebo is reportedly effective in treating the disease. The disease is mild and lack of treatment is not considered to be medically important or hazardous. No standard treatment exists. We can give placebo when there is a no standard treatment exists or standard treatment is ineffective. Then only we need to give placebo. There are again certain designs like parallel group design. It is a group comparison. Parallel design means 2 to 3, 4 multiple groups are there. Then we can give different intervention or same intervention to different variety of participants to check the results, or we can give different interventions too. Then matched pair parallel design. In this method, pairs of subjects are formed and possessing the same characteristics and who might be expected or respond similarly to the treatment. And complete randomized design, in which each patient receives one and only one treatment in a randomized fashion. Group comparison. The advantages are it's simple, most common, and easy to implement. It is universally acceptable. It is applicable to acute conditions. Analysis is less complicated and interpretations is straightforward. Disadvantages are it does not uh, come into account with the individual variability. If multiple groups to be compared, then not comes under gold standard, because RCT requires only mainly two groups. RCT as randomization is not possible in multiple groups. So as per WHO, a clinical trial is any research study that prospectively assign human participants or group of humans to one or more health related interventions to evaluate the effects on health outcomes. What is the need of RCT? It is a more organized research. Multicentric researchers are possible. Data based on research outputs. Central registry, that is CTRI, of research works, so everyone could know about our work. More intensive training for research personnel or core issues of Ayurveda, that is Prakriti analysis, Deha, Rog, Agni, Sattva, Bal analysis, etc. standardization of drugs or modalities of treatment or record system is needful. So whenever we do in our RCTs, we must take the Ayurveda concepts also. We need to involve Ayurveda concepts also. Factors in choosing clinical trial designs. Chronology of events. Chronological effects may be very important in any trial design. Means what is the chronicity of symptoms, and accordingly uh, we need to check the uh, symptoms and outcome. But particularly in crossover design, what are the result of intervention A, and after a washout period, and then we need to check the intervention B. So which one is better, we need to guess, we need to calculate. Then subject convenience, because the trials are RCTs are lengthy, and so we need to check the convenience of participants too. Their conveyance, many times we need to give some incentives to facilitate their timely arrival at a research place. So uh, so many factors are important while designing the CT, that is clinical trial. We need to take financial concern, that is trial cost, and budget must be, if budget is much more, then we need to get fund or help from other institutes, then only it is feasible. Now the RCTs are being done on different phase wise trial. That is, phase 1 means all patients receive the drug, and unblinded state open label trial is suitable here, because it is only few less than 50 or just before 30 to 50 or few patients are enrolled to check the efficacy mainly. In phase 2, 2, 3, parallel design or crossover or factorial design are most commonly used. And phase 4, 4, non-experimental or observational designs is suitable. These include epidemiologically or ecologically designs such as case control or cohort studies. So phase 1 means we need to check the endpoints as the objectives as a safety and acceptability, and the size is up to 50 only, and they are healthy volunteers. They should not be diseased, or if we have to take diseased one, then the patients must be very less, below 30. Then phase 2, long term safety we need to check, and dose or schedule frequency need to check by this assigning to few hundreds, like 100 to 500 is the total size of participants, and the risk is always low. Then in phase 3, from few hundreds to few thousands is the size, and the risk is involved is high, and we need to check here the effectiveness of a intervention. Then in phase 4, we need to check the from few thousands to lakhs of people as a community based trial to work as a post marketing surveillance. Phase 1 we already discussed, phase 2, phase 3, phase 4, and in phase 4, suppose some rare side effects may only be found in large groups of people, because already we have undertake in phase wise manner. So if there is any issue or any side effect or any adverse reaction, then we can come across. There are certain guidelines whenever we conduct the research, then these guidelines are very helpful to conduct it properly. So this is a www.consortstatement.org. This is the site, and this is a checklist. This is the checklist. This is the checklist in which we can find so many things. It is as a presenter mode, it is not able to open it. On net, you can check consort guideline. It is available in on this website. I think yeah, it is open, and this is the checklist in which we need to check methods, trial design, then sample size, its outcome, inclusion and exclusion criteria, allocation, randomization method, sequence, concealment, and so many issues are there. And it is a guiding line that how you have to go through it, or how sample size was determined, or what is the way, what is the method of randomization or allocation, like that. This is a useful guideline in which whenever we have to conduct RCT, this guideline is very, very useful. Observational studies. It is a strobe guideline and elixir of guidelines, strobe checklist, cross-sectional. This is a different guidelines in which mainly strobe initiative is available at www.strobestatement.org or epidem.com. You can go through it on net. On again, there is a third design, spirit design for study protocol. Whatever synopsis or protocol we need to prepare as per Prisma or Spirit guideline, we have to check it thoroughly, and accordingly we have to add the things. If we are missing it, then we need to add as per the checklist. For clinical practice guidelines, agree guideline is there. Reporting guideline checklist. It is a checklist in available on etiquette on Equator Network. Now coming to the do's and don'ts. Do's and don'ts. What to do and what not to do. Do's for conducting research are protect the confidentiality, care and support. Because if someone is leaking that their confidentiality, and even the participants always want that even their family members should not know about their uh, involvement in research study as a participant. So we need to keep their to maintain their confidentiality, and whenever they seek any help or care, or we must provide them support. For ensuring the transparency, there is a new tool as an audio visual recording where during informed consent process to make the transparency at most. So there will be no any as such manipulation or any data mishandling. So ensure the safety prior to RCT by animal experiments or QA/QC, that is quality assurance and quality control, toxicity study, and GCP, that is good clinical practice. Then if they are suffering with any kind of adverse drug effect or any hazard or any risk, then we need to give compensation as per policy and as per the calculation of DCGI. Then whenever we have to conduct the result on a multi-centric basis, then we need to standardize our protocol, study protocol. Um, After standardization only, we can undertake multi-centric trials. And we should facilitate incentives to participants if they are uh, demanding the conveyance or any because they are losing with their daily wages or their time is getting waste, and so many issues are there. So we have to face so many challenges, accept the challenges and go forward with devotion, dedication and discipline or determination. So these 4Ds are very much important while conducting the research. We need to keep patience because these trials are so much troublesome many times because patients are not cooperating. If participants are not cooperating, then we need to provide incentives also to facilitate our research and go forward with devotion, dedication and discipline. What are the don'ts? Don'ts are dilution of the guidelines, acceptance of biases and errors. We should not accept these biases and errors. Conduction with IC and CTRI non-approval. If CTRI and IC is not approved the synopsis or protocol, then uh, we should not go forward as a to start the research. Until and unless we don't get approval, then we can't start the research. Non-approval of regulatory board if the trial is international trial. Deny to DCGI or GEAC when trial is based on genetics. Uh, Based on genetics, then we need to take approval from GEAC. And if it is a society based, then we need to take the approval from Health Ministry screening committee. And we need to inform time to time about any ADR, that is adverse drug reaction, to regulatory board also and to IC and as well as sponsor. And the non-informal information to them, it acts as a don'ts. So at the end, summary, to summarize the things, the ultimate goal of a clinician is to help patients live long and disease free. By adopting the proper research design, one can conclude with latest facts, update the science to maximize patient care. So research conduction as per different guidelines described on Equator Network, that is consort, enhancing the quality and transparency of health research as per good clinical practices. Its goal is to protect the rights, safety and welfare of human participating in research and to assure the quality and reliability of data collected without manipulation of outcomes. And conclusion is proof to concept or to precede clinical trials. The design must be very robust and with inclusion of Ayurvedic concepts. Objective to evaluate treatment regimen including the trial, assessment of quality of lifestyle and disease parameters. Articles to be published as per peer-reviewed high-index journals like Q1 to Q4. Then the all over the globe, the demand is high, and there is demand of RCT and demand of highly reputed high-index journals. If we conduct properly, if we do conduct the proper research by putting proper research design, the research outcomes must be published in a good index, high index journals, and the references must be as per the guidelines of standard guidelines as per International Committee of Medical Journal Editors, ICMJE. compliance of GCP or GAP, GMP, that is good medical practice, and GEP and ICMR guidelines. Proper description of SOPs of the trial drugs, consistent quality of products, and sincere efforts to improve the quality of clinical trials. Evaluation of data by using biostatistical tools and interpretation by Ayurveda are very much important, and it is a need of the hour. So this is all for the topic of RCT. Thank you all for patient listening. Any queries, please feel free to ask. Thank you one and all.
Thank you, ma'am. It was really a very nice presentation on RCT and its type. Now move to question answer round. To all participants, if anyone having any doubt, he or she can ask. Anyone having any doubt?
I will be glad to answer your questions. You can see here some query. What is quadruple blending and what is double dummy design? Double dummy design is not practical in Ayurveda setup. So only thing is that we need to keep thinking on only double blind, because triple blind is also not feasible, because many times institute don't have biostatistician, and it is not much feasible. So double dummy is not practically possible. And what is quadruple blinding? It is again not possible for Ayurveda setup. Many times it is by very with very difficulty only double blind is possible, and in certain trials only we can do triple blinding. Quadruple blinding is not actually, it is just mentioned but no elaboration is there. Anything else? Agri guideline is for diagnostic research or clinical practice. Like GCP, good clinical practices, it is helpful. Agree.
Okay, now we conclude the session after the vote of thanks. Myself, Dr. Bhavna Bhardwaj, PG Scholar of Panchakarma Department of Ayurveda and Unani Tibia College and Hospital. I would like to thank our guest speaker, Dr. Renu Rathi ma'am, for enlightening us with their knowledge about randomized clinical trials and its types. Today's webinar was full of knowledge about the clinical trials like randomization, quasi-trial, and etc. It provides some interesting points. And lastly, thanks to all participants from behalf of Panchakarma department for patiently listening this webinar.
Thank you. Thank you, everyone.
Thank you, ma'am. Next session on Monday,
Yes.
ma'am will continue the session on single case design, case control studies, ethnographic study, black box design, crossover design, factorial design, and phases of clinical studies on Monday at 2 p.m. to 3:30 p.m. Thank you, ma'am. Thank you, everyone.
Thank you.